Welcome to this tutorial on plasma membrane and its functions, brought to you by Medical Biochemistry, Power Capsule Series. This video pertains to the following CBME curriculum topic. Describe the molecular and functional organization of a cell and its subcellular components. This tutorial is preceded by Tutorial 1A, and we therefore suggest that you should also go through Tutorial 1A, which is available in this series. The plasma membrane is a selectively permeable membrane that separates the cell from the external environment through regulating the entry and exit of molecules to and from the cell. The plasma membrane ensures that different types of cells perform their specialized functions in a protected and unique environment. Ever since Singer and Nicholson described the double-layered fluid mosaic model of the plasma membrane, a lot of progress has been made, and now we have a plethora of information about the organization and function of this important structure. Far from being a static and inert barrier, the membrane actually is very dynamic and performs plenty of functions depending on the cell type. These functions vary. The plasma membrane is fluid in character and is a continuous double layer comprising of two monolayers, which consist of amphipathic lipid molecules in which cholesterol and membrane proteins are embedded. The two layers have dissimilar molecular composition and function. The plasma membrane comprises primarily of phospholipids, cholesterol, glycolipids and proteins. Proteins and lipids comprise approximately 50% each, although it varies from cell to cell. Lipids provide the basic structure of the bilayer. Individual molecules can rapidly diffuse and move around within their own monolayer. The major lipids in the plasma membrane belong to the phospholipid class: phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine. Sphingomyelin and inositol phospholipids are the phospholipids found. Phosphatidylserine is negatively charged. The bipolar nature of phospholipid molecules, with a polar head and nonpolar hydrophobic hydrocarbon tail, causes lipid molecules to spontaneously aggregate and bury their hydrophobic tails in the interior, and to expose their hydrophilic heads towards aqueous medium. This behavior is due to the shape and amphipathic nature of the phospholipid molecules, and makes it possible for the bilayer to form spontaneously, and is fundamental to the creation of a living cell. In this animation, you can see that hydrophobic tails naturally tend to face away from water molecules with hydrophilic head groups aligned towards water. The membrane is fluid in character, and this fluidity is functionally important. Membrane fluidity has to be precisely regulated because it affects transport processes and enzyme activities. When fluidity decreases, rigidity increases. Presence of double bonds in the hydrocarbon tails increase fluidity. Double bonds in C's configuration favors fluidity. The reason is that it is more difficult for the tails to tightly pack together when unsaturated fatty acids, particularly in C's configuration, are present, and therefore the fluidity increases. In general, cholesterol decreases fluidity and makes the bilayer less deformable, which affects permeability and other functions of the membrane. Cholesterol enhances the permeability barrier properties of the lipid bilayer. However, the effect of cholesterol on membrane fluidity is temperature dependent. Cholesterol is almost absent in bacterial membranes. Now, moving on to the stars of the show, let's discuss membrane proteins. These are the key players in the membrane's functionality. Whereas the lipid bilayer determines the basic structure of biological membranes, proteins are responsible for most of the specific functions of membrane. Membrane proteins are proteins that are embedded in or attached to the cell membrane. The proteins give each type of membrane in the cell its characteristic functional properties. The amounts and types of proteins in a membrane are highly variable. Integral membrane proteins can be of two broad types: transmembrane proteins and lipid-anchored proteins. The firm attachment of integral proteins to membranes is the result of hydrophobic interactions between membrane lipids and hydrophobic domains of the protein. Transmembrane integral proteins span the entire thickness of the cell membrane. Peripheral proteins, on the other hand, are loosely attached on the surface of one monolayer, more on the non-cytosolic monolayer, and are involved in cell signaling, cell adhesion, and enzymatic activity. Understanding the functions of the membrane proteins is not only essential for biology enthusiasts, but also for those exploring medical applications and treatments. A huge range of pharmaceutical molecules work by targeting the membrane proteins. Let us now look at some of the functions of membrane proteins. Very few molecules can just diffuse through the membrane, and most molecules require help in order to enter or to come out. Transportation of molecules and ions across the membrane is an important function of membrane proteins. Most of these proteins can be highly selective, letting through only specific molecules or ions. Certain membrane proteins have enzymatic functions, and they catalyze chemical reactions on or near the membrane. Enzymes required for lipid and carbohydrate biosynthesis and catabolism are just a couple of examples. You may also check these other well-known membrane enzymes. Membrane proteins play a crucial role in the structural integrity of the cell membrane, ensuring that it retains its proper shape and integrity. Proteins that help connect the cytoskeleton to membranes are an example. Membrane proteins have the ability to identify and bind to foreign molecules or ions like antibodies or antigens. The immune system component relies on these interactions. This also helps distinguish between self and non-self molecules, and therefore has a role in various immunological responses and hypersensitivity reactions. Signaling molecules like hormones or neurotransmitters can attach to specific membrane proteins that function as receptors. This generates cellular response through a process called signal transduction. Such cellular responses include alterations in the function of other membrane proteins, activation of intracellular signaling pathways, changes in gene expression, etc. Cell adhesion is the process by which cells adhere to one another or to the extracellular matrix, and membrane proteins can play a part in this process. Tight junctions and desmosomes are two examples of cell-cell junctions that may involve these proteins. Ion channels are a category of membrane proteins that open up under specific circumstances and let ions pass through. The permeability of these channels to ions can be controlled in a number of ways, making them selective. Ion channels are an important part of the cell membrane transport mechanism. Small non-polar molecules readily dissolve in lipid bilayers and therefore diffuse rapidly across them. Small uncharged polar molecules also diffuse across a bilayer. But lipid bilayers are highly impermeable to most polar molecules and ions of biological importance, and therefore special mechanisms are required to transport molecules that cannot cross the membrane barrier. In general, the smaller the molecule and the more hydrophobic or nonpolar it is, the more rapidly it will diffuse across a lipid bilayer. By contrast, lipid bilayers are highly impermeable to hydrophilic molecules, ions and large molecules. The charge and high degree of hydration of such molecules prevents them from entering the hydrocarbon phase of the bilayer. Therefore, there exists various transport mechanisms by which molecules can move in and out of cell, and many of these mechanisms are made possible through membrane proteins. The various mechanisms can be categorized under any of the two broad categories. So, we have passive transport, and we have active transport mechanisms. Passive transport occurs spontaneously down an electrochemical gradient, either by simple diffusion through the lipid bilayer or by facilitated diffusion through channels or passive carriers. By contrast, active transport requires an input of metabolic energy and is always mediated by carriers that utilize metabolic energy to pump the solute against its electrochemical gradient. Passive transport of molecules occurs spontaneously down an electrochemical gradient. Input of energy is not required in the animation. We can observe the various types of passive transport mechanisms in simple diffusion. We can see the red molecule simply passing through the membrane. Another molecule is freely passing through the yellow-colored channel. Yet another molecule, which may also be an ion, is passing through a channel that is controlled through a gated mechanism. This gated mechanism responds to a ligand, voltage changes, etc. Finally, we also observe a molecule being transported by a carrier protein, which is not a channel. The carrier protein changes its configuration in order to allow the passage of the molecule. None of the above mechanism requires any direct input of energy. Simple passive diffusion requires no additional energy source other than what is found in the solute's electrochemical or concentration gradient. Solute reaches equilibrium across the membrane. The solute crosses the membrane anywhere by simply dissolving into ion diffusing through the lipid bilayer. Facilitator is not required. In facilitated diffusion, solutes cross the membrane at a specific location where diffusion is assisted by solute-specific proteins that act as facilitators, which may be a channel or a carrier. Facilitators enable specific hydrophilic solutes to cross the membrane without coming into direct contact with the hydrophobic interior of the lipid bilayer. Facilitators can recognize their solute with precision. Facilitated diffusion is much faster than simple diffusion. Active transport requires energy, often in the form of ATP, and results in a non-equilibrium and a net accumulation of the solute on one side of the membrane. In active transport, special transport proteins actively pump certain solutes across the membrane against their electrochemical gradient. The pumping activity of the carrier protein is directional and is tightly coupled to a source of metabolic energy, such as ATP hydrolysis or an ion gradient. Active transport can be of two types: primary active transport and secondary active transport. Primary active transport is also called direct active transport. It involves using energy to directly pump a solute across a membrane against its electrochemical gradient. Secondary active transport systems are composed of two separate functions. The energy-dependent movement of an ion generates an electrochemical gradient of the ion across the membrane. This ion gradient is coupled to the movement of a second solute in either the same direction or in the opposite direction. Movement of the second solute down its electrochemical gradient is by facilitated diffusion. Secondary active transport by sodium-glucose transport protein 1 in the intestinal epithelium is an example. SGLT1 uses the energy in a downhill transmembrane movement of sodium to transport glucose across the apical membrane against an uphill glucose gradient, so that this glucose can be transported into the bloodstream. Passive or active transport can be of any of the following types. The uniport system carries a single ion or molecule across the membrane. In the cotransport system, the transfer of one molecule depends on the simultaneous or sequential transfer of another molecule. In symport cotransport system, the transport protein carries to different ions or molecules in the same direction. And in antiport cotransport system, the transporter carries to molecules or ions in the opposite direction. Hence, we have come to know that there are three main categories of transmembrane transport proteins. Channel proteins, that are required in passive transport facilitated diffusion. Carrier proteins that are required in passive transport facilitated diffusion. And also, carrier proteins that are required in active transport. The most important active transport protein is the plasma membrane-bound sodium-potassium pump. This single enzyme accounts for 1/3 of human energy expenditure and is often referred to as the pacemaker for metabolism. Apart from passive transport and active transport, there are several other ways of transport. Large macromolecules can cross membranes by these other methods. These methods involve large sections of a membrane containing many lipids and proteins. We have receptor-mediated endocytosis, phagocytosis, pinocytosis, and exocytosis. Membrane blebbing. In endocytosis, the plasma membrane extends and folds around desired extracellular material, forming a pouch that pinches off, creating an internalized vesicle. In receptor-mediated endocytosis, the molecule that is required to be taken up into the cell binds with a specific receptor, and the entire receptor molecule complex is then internalized. After internalization, the receptors are released back to the surface. Pinocytosis is a form of endocytosis involving fluids containing many different substances. This process occurs in cells lining the small intestine and is used primarily for absorption of fat droplets. The invaginated pinocytosis vesicles are much smaller than those generated by phagocytosis. The vesicles eventually fuse with lysosomes, whereupon the vesicle contents are digested. Phagocytosis is a type of endocytosis that involves uptake of large solid particles. The particles can be aggregates of macromolecules, parts of other cells, and even whole microorganisms. In phagocytosis, surface proteins may specifically recognize and bind to the solid particles. Phagocytosis generates intracellular vesicles, called phagosomes, that have sequestered solid particles for transportation to lysosomes for digestion. Exocytosis is the process by which cells release large molecules from the cytoplasm to the cell exterior. It is the opposite of endocytosis. Exocytosis generates vesicles, referred to as secretory or transport vesicles. These intracellular secretory vesicles fuse with the plasma membrane and release their aqueous, sequestered contents to the outside, simultaneously. The vesicular membrane hydrophobic components, such as lipids and proteins, get added to the plasma membrane. Blebbing of the plasma membrane is a morphological feature of cells undergoing late-stage apoptosis, that is, programmed cell death. A bleb is an irregular bulge in the plasma membrane of a cell, and this bulge eventually blebs off from the parent plasma membrane, taking part of the cytoplasm. Gap junctions are a structural feature of many animal plasma membranes that allows transmembrane passage of small solutes like ions, sugars, amino acids, and nucleotides, while preventing migration of organelles and large polymers like proteins and nucleic acids. Gap junctions connect the cytoplasm of two adjacent cells through non-selective channels. Each channel in a gap junction is made up of 12 proteins called connexins. Gap junctions are particularly important in cardiac muscle, as the electrical signals for contraction are passed efficiently through these channels. Malfunctions of gap junctions lead to a number of human disorders, including demyelinating neurodegenerative diseases, skin disorders, cataracts, and even some types of deafness. Thank you for joining us today on Medical Biochemistry Power Capsule series. As we wrap up our journey through the plasma membrane, it's clear that this structure is not just a passive barrier. It's a dynamic and vital player in the cellular orchestra. We've only scratched the surface, but I hope this video sparks your curiosity to explore further.
Our next tutorial in the Medical Biochemistry Power Capsule series is titled, Applied and Clinical Concepts Related to Membranes and Transporters. The tutorials presented in the MBPC series are not intended to be a substitute for your textbooks. However, we feel that these tutorials can prove to be very valuable as complementary educational aids and for purpose of rapid revision prior to exams. If you enjoyed this video, don't forget to hit the like button, subscribe for more exciting content, and ring that notification bell to stay updated. Until next time, keep exploring the wonders of medical science.